Speaking about BBYCT 131. So in this, we'll be having about biodiversity. So we'll be discussing about microbes, algae, fungi, and archegoniates. First, we'll discuss about viruses. Viruses, ವೈರಸ್ಸುಗಳು ಅದನ್ನ ಹೇಗೆ ಡಿಫೈನ್ ಮಾಡ್ತೀವಿ ನಾವು viruses, it can be defined as small obligate intracellular particles which can be seen only by electron microscope and that infect and take over the host cell to replicate itself. Now, there is a controversy. Viruses are living or non-living. ವೈರಸ್ಸುಗಳು ಜೀವಿಗಳ ಅಥವಾ ನಿರ್ಜೀವಿಗಳ. So this controversy, it started since TMV was first crystallized. So why viruses are considered living? ವೈರಸ್ಸುಗಳನ್ನ ಜೀವಿಗಳು ಅಂತ ಯಾಕೆ ಹೇಳ್ತೀವಿ? Because they possess genetic material, DNA or RNA, and protein. They can replicate only inside the host cell. They use the cellular machinery of the cells like ribosomes, tRNA, enzymes, etc., to replicate its genome and then synthesize a protein code. Even these viruses can be crystallized. The crystallized virus, they conserve the living properties. And they are transmitted. ಅದು ಹೇಗೆ ಹರಡುತ್ತೆ ಅಂದರೆ ಸೋಂಕಿತರಿಂದ ಸೋಂಕಿಲ್ಲದವರೆಗೆ ಹರಡುತ್ತೆ ಅದು. So why ವೈರಸ್ಗಳು ನಿರ್ಜೀವಿ ಅಂತ ಹೇಳ್ತೀವಿ? Because they are unable to show living properties outside a host cell and they can be crystallized. Now, viruses can replicate inside host but cannot grow. They do not have the metabolic machinery. Now, phases ಬಗ್ಗೆ ಮಾತಾಡೋಣ. Phases, viruses ಅಲ್ಲಿ ಎರಡು phases ಇರುತ್ತೆ. One is the extracellular and the other is intracellular. In extracellular phase, the complete virus particle is called viron. Complete virus again ಹೇಳ್ತೀವಿ viron ಅಂತ ಕರಿತೀವಿ, which remains in a inert state. That is, it does not carry out any metabolic activity. ಮತ್ತೊಂದು phase ಬಂದ್ಬಿಟ್ಟು intracellular phase. ಈ phase ಅಲ್ಲಿ the virus replicates by using genome and metabolic machinery of the host cell. Now, the viruses, as it cannot multiply outside the host cell, has considerable medical importance for the development of antiviral drugs. Now, antiviral drugs, as the name it indicates, which means the drugs which destroy the viruses. Now, coming to the structure, ಈಗ ರಚನೆ ಬಗ್ಗೆ ಮಾತಾಡೋಣ. So, ವೈರಸ್ಸುಗಳು, these are made up of only ಎರಡೇ components. ಆ components ಏನಂದ್ರೆ ಒಂದು ಪ್ರೋಟೀನು ಮತ್ತೊಂದು ನ್ಯೂಕ್ಲಿಕ್ ಆಸಿಡ್. So viruses, they are made up of only two components, protein and nucleic acid. That is the viral genome. So the viral genome is protected against chemical and physical damage by the protein coat of the virus, which is called as a capsid. The protein coat of the virus particle is called as capsid. So the capsid and the viral genome together are called the nucleocapsids. So the building blocks of the capsids are proteins, which are called as capsomeres. So on the viral capsid, special capsid proteins are there, which are called spikes. So these spikes are present on the binding sites of the virus so that it can attach to the specific receptors of the host cells. So these capsomeres, that is the protein subunits, they are responsible for three different symmetry groups of viruses. We have three different symmetries. One is the icosahedral, the second is helical, and the third is the complex. So here there is a picture of the helical virus. So what is happening here? In this helical virus, the capsid or the protein coat, it is arranged in the form of a filamentous form or a rod-shaped structure. So in this rod-shaped structure, you have a cavity. So this cavity encloses your viral genome. So example for this will be a tobacco mosaic virus, which is a helical virus. So based on the symmetry, I told you three groups are there: icosahedral, helical, and complex. So helical example is tobacco mosaic virus. Next, coming to the second symmetry, that is icosahedral. So in icosahedral, the viral capsid, it has similar subunits. So these subunits are arranged in a equilateral triangle. And these are arranged in a proper symmetry. Next is about example for this will be adenovirus. Example ಕೊಡ್ಬೋದು ನಾವು ಇದಕ್ಕೆ. ಇನ್ನೊಂದು ಲಾಸ್ಟ್ ಸಿಮಿಟ್ರಿ ಬಂದ್ಬಿಟ್ಟು ಕಾಂಪ್ಲೆಕ್ಸ್ ಸಿಮಿಟ್ರಿ. ಸೊ ಈ ಕಾಂಪ್ಲೆಕ್ಸ್ ಸಿಮಿಟ್ರಿ ಹೆಲಿಕಲ್ ಮತ್ತೆ ಐಕೋಸಾಹೆಡ್ರಲ್ ಸಿಮಿಟ್ರಿಗೆ ಸೇರಲ್ಲ ಇದು. ಯಾಕಂದ್ರೆ ಇದಕ್ಕೆ ಸ್ವಲ್ಪ ಅಡಿಷನಲ್ ಸ್ಟ್ರಕ್ಚರ್ಸ್ ಇರುತ್ತೆ. ಬಿಕಾಸ್ it can have a tail plate irbodu, tail fibers irutte, the complexity irutte. So this comes under separate group called as complex symmetry. So idik example now bacteriophage kodabodu. So this is about the symmetry groups of viruses: icosahedral, helical and complex. Icosahedral example banbitu adenovirus, helical ge example TMV kodabodu, ಮತ್ತೆ complex ge bacteriophage example ಕೊಡ್ತೀವಿ. Now in some viruses, by convention, the icosahedral ಮತ್ತೆ helical structure viruses ಅಲ್ಲಿ, the nucleocapsid, it is surrounded by a flexible, loose fitting membrane which is made up of proteins ಮತ್ತೆ lipids in the made ಆಗುತ್ತೆ, and these are acquired from the host cell during replication. So that loose fitting membrane is known as envelope. So virus, if they have envelope, they are called as enveloped viruses. ಇಫ್ ವೈರಸ್ಗಳಲ್ಲಿ ಈ ಎನ್ವಿಲೋಪ್ ಇಲ್ಲ ಅಂದ್ರೆ ಅದನ್ನ ನಾವು ನೇಕೆಡ್ ವೈರಸಸ್ ಅಂತ ಹೇಳ್ತೀವಿ, ಅಥವಾ ನಾನ್ ಎನ್ವಿಲಪ್ಡ್ ವೈರಸಸ್. ಈಗ ನ್ಯೂಕ್ಲಿಕ್ ಆಸಿಡ್ ಅಂತ ಹೇಳಿದೆ, ವೈರಸಸ್ ಅಲ್ಲಿ ದ ನ್ಯೂಕ್ಲಿಕ್ ಆಸಿಡ್ ಆರ್ ದ ಜೀನೋಮ್, ಅದು ಬೇಕಿದ್ರೆ DNA ಆಗಿರ್ಬೋದು ಅಥವಾ RNA ಇರ್ಬೋದು. ವೈರಸ್ಗಳಲ್ಲಿ DNA ಇದ್ರೆ ಅದನ್ನ DNA ವೈರಸ್ ಅಂತ ಕರಿತೀವಿ. RNA ಜೆನೆಟಿಕ್ ಮೆಟೀರಿಯಲ್ ಇದ್ರೆ ಅದನ್ನ RNA ವೈರಸಸ್ ಅಂತ ಹೇಳ್ತೀವಿ. So RNA ಮತ್ತು DNA viruses, they are further classified into 7 classes. So we have a Baltimore classification. So as you see here on the screen, we have 7 classes ಇದೆ ಅದರಲ್ಲಿ. ಫಸ್ಟ್ ಕ್ಲಾಸ್ ಬಂದ್ಬಿಟ್ಟು ಡಬಲ್ ಸ್ಟ್ರಾಂಡೆಡ್ DNA genome. ಎಕ್ಸಾಂಪಲ್ ನಾವು ಲ್ಯಾಂಬ್ಡಾ ಫಾಜ್ ಕೊಡ್ಬೋದು, ಲ್ಯಾಂಬ್ಡಾ T4. In case of animal viruses, we have herpes virus, pox virus, ಅದು ಎಕ್ಸಾಂಪಲ್ ಕೊಡ್ಬೋದು. ಸೊ ಸೆಕೆಂಡ್ ಕ್ಲಾಸ್ ಬಂದ್ಬಿಟ್ಟು ಸಿಂಗಲ್ ಸ್ಟ್ರಾಂಡೆಡ್ DNA genome ಸಿಂಗಲ್ ಸ್ಟ್ರಾಂಡೆಡ್ ಇರುತ್ತೆ. ಸೊ ಇದಕ್ಕೆ ಎಕ್ಸಾಂಪಲ್ 5X174, ಅದು ಬ್ಯಾಕ್ಟೀರಿಯಲ್ ವೈರಸ್ ಎಕ್ಸಾಂಪಲ್. ಇನ್ನೊಂದು ಬಂದ್ಬಿಟ್ಟು ಚಿಕನ್ ಅನಿಮಿಯಾ ವೈರಸ್, ದಟ್ ಎಕ್ಸಾಂಪಲ್ ಅನಿಮಲ್ ವೈರಸ್. ಥರ್ಡ್ ಕ್ಲಾಸ್, ಮೂರನೇ ಕ್ಲಾಸ್ ಬಂದ್ಬಿಟ್ಟು ಡಬಲ್ ಸ್ಟ್ರಾಂಡೆಡ್ RNA genome. ಸೊ 5-6 ಎಕ್ಸಾಂಪಲ್ ಇದೆ, 5-6 the symbol is that. So for in case of animal viruses, example Rio virus. Then class 4 is single stranded RNA genome with a plus sense, example MS2 ಮತ್ತೆ polio virus example ಕೊಡ್ಬೋದು ಇದಕ್ಕೆ. Next fifth class ಬಂದ್ಬಿಟ್ಟು single stranded RNA genome with minus sense. Okay, so example ಇದಕ್ಕೆ ಬಂದ್ಬಿಟ್ಟು influenza virus, rabies virus, ಅದು example ಕೊಡ್ಬೋದು ನಾವು. Sixth class ಬಂದ್ಬಿಟ್ಟು single stranded RNA genome. So this replicates with a DNA intermediate in the single stranded RNA genome ಬರುತ್ತೆ. So example ಬಂದ್ಬಿಟ್ಟು retrovirus. Class 7, the last class, double stranded DNA genome that replicates with RNA intermediate. Double stranded DNA genome with RNA intermediate. Example ಬಂದ್ಬಿಟ್ಟು hepatitis B virus. So ಇದು classification. So virus na DNA ಮತ್ತೆ RNA viruses we can classify into 7 groups. Now coming to the importance, these viruses are economically important as they cause losses from diseases, and also it plays a major beneficial role in the medicine research as well as in diagnostics. So medicinally, vaccine production. So viruses are used in vaccine production. So vaccine is produced using viruses. It can be produced by killing the virus or by attenuated virus. You can produce the vaccine. Then gene therapy means if there is a defective gene, so that gene can be replaced by a functional gene. So that is gene therapy. Then cancer therapy, even cancer viruses are used for production of anti-cancer drugs. Then viral biopesticides. So viral biopesticides, they are least significant compared to bacterial biopesticide, but baculovirus, it is a very good example for the viral biopesticides. So this is about the importance. Next, coming to the replication. So replication means here virus, how it is multiplying. ಅದು ಹೇಗೆ multiply ಆಗುತ್ತೆ. So viral replication results in many progenies, many copies produce ಆಗುತ್ತೆ, and these progenies, they leave the host cell so that it can infect other cells in the organisms or spread the infection. So viruses, as it is replicating, ಸೊ ರೆಪ್ಲಿಕೇಶನ್ ಯೂಶಲಿ 5 steps ಅಲ್ಲಿ ಇರುತ್ತೆ. 5 steps. ಫಸ್ಟ್ ಸ್ಟೆಪ್ ಬಂದ್ಬಿಟ್ಟು ಅಟ್ಯಾಚ್ಮೆಂಟ್. ಆಸ್ ಯು ಕ್ಯಾನ್ ಸಿ ಹಿಯರ್ ಇನ್ ದ ಪಿಕ್ಚರ್, ಫಸ್ಟ್ ವೈರಸ್ ಏನಾಗುತ್ತೆ ಅದು ಬಂದ್ಬಿಟ್ಟು ಹೋಸ್ಟ್ ಸೆಲ್ಗೆ ಅಟ್ಯಾಚ್ ಆಗುತ್ತೆ. ಸೊ ರಿಸೆಪ್ಟರ್ಸ್ ಇರುತ್ತೆ, ಥ್ರೂ ರಿಸೆಪ್ಟರ್ಸ್ ಇಟ್ ವಿಲ್ ಕಮ್ ಅಂಡ್ ಅಟ್ಯಾಚ್ ಟು ದ ಹೋಸ್ಟ್ ಸೆಲ್. ಅದು ಫಸ್ಟ್ ಸ್ಟೆಪ್, ಬಿಕಾಸ್ ವೈರಸ್ ರೆಪ್ಲಿಕೇಟ್ ಮಾಡೋದು 5 steps ಪ್ರಾಸೆಸ್ ಅದು. ಸೊ ಫಸ್ಟ್ ಪ್ರಾಸೆಸ್ ಬಂದ್ಬಿಟ್ಟು ಅಟ್ಯಾಚ್ಮೆಂಟ್. ವೈರಸಸ್ ಬಂದ್ಬಿಟ್ಟು ಹೋಸ್ಟ್ ಸೆಲ್ಗೆ ಅಟ್ಯಾಚ್ ಆಗುತ್ತೆ ಡ್ಯೂ ಟು ಸಮ್ ರಿಸೆಪ್ಟರ್ಸ್ ಇಂದ, ಸೊ ದಟ್ ಇಟ್ ಕ್ಯಾನ್ ಕಮ್ ಅಂಡ್ ಬೈಂಡ್ ಟು ದ ಸರ್ಫೇಸ್ ಆಫ್ ದ ಹೋಸ್ಟ್ ಸೆಲ್. ಸೊ ಒನ್ಸ್ ಅದು ಬಂದ್ಬಿಟ್ಟು ಬೈಂಡ್ ಆಗಿದ್ಮೇಲೆ ಸೆಕೆಂಡ್ ಸ್ಟೆಪ್ ಏನಾಗುತ್ತೆ, ಪೆನಿಟ್ರೇಷನ್. ದ ವೈರಸ್ ಒಳಗೆ ಹೋಗ್ಬೇಕು ಹೋಸ್ಟ್ ಸೆಲ್ ಒಳಗೆ, ಅದು ಪೆನಿಟ್ರೇಷನ್ ಸ್ಟೆಪ್. ಸೊ ವೈರಸ್ ಹೋಸ್ಟ್ ಸೆಲ್ ಒಳಗೆ ಹೋಗುತ್ತೆ, ದಟ್ ಇಸ್ ದ ಸೆಕೆಂಡ್ ಸ್ಟೆಪ್, ಪೆನಿಟ್ರೇಷನ್. ದ ಥರ್ಡ್ ಸ್ಟೆಪ್ ಇಸ್ ಅನ್ಕೋಟಿಂಗ್. ಅಲ್ಲಿ ಹೋಗ್ಬಿಟ್ಟು ಪ್ರೋಟೀನ್ ಕೋಟ್ ಏನಿದೆ ವೈರಸಸ್ ಅಲ್ಲಿ ಅದೆಲ್ಲ ಅನ್ಕೋಟ್ ಆಗುತ್ತೆ. ಅನ್ಕೋಟ್ ಆದ್ಮೇಲೆ ದ ಪ್ರೋಟೀನ್ ಕೋಟ್ ಇಸ್ ರಿಮೂವ್ಡ್ and you get naked nucleic acid. This nucleic acid, it enters the cells. Naked nucleic acid enters the cell. That is third step, uncoating step. Next, fourth step ಬಂದ್ಬಿಟ್ಟು biosynthesis. Biosynthesis ಅಲ್ಲಿ ಏನಾಗುತ್ತೆ, the viral genome ಏನಿದೆ ಅದು replicate ಆಗುತ್ತೆ. It will take up the host cell machinery and it starts replicating. So it synthesizes nucleic acids and nucleic acid is replicated and then proteins are synthesized. So here biosynthesis step ಆಗುತ್ತೆ. That is the fourth step here. Next, coming to the last step, once nucleic acid synthesize agide ಮತ್ತೆ protein synthesize agide ಅದನ್ನ organize ಮಾಡಬೇಕು. ಅದು it has to assemble. So the virus is assembled. That is our last step, assembly and release. So the virus will assemble into a complete virus, and then virus particles are ready, so it has to release. Adu hege release agutte, so it will kill the cell, the host cell. It will lyse the host cell and the virus particles are released out so that it is ready to infect the other cell. So this is how the replication is taking place. Replication 5 steps agutte. First step banbitu attachment ali the virus will attach to the host cell. After attaching enagutte, a viral particle enter agutte, penetration agutte. That is second step, the virus has to enter. Third step, uncoating. Matlab protein matte nucleic acids, protein is removed and nucleic acid enters the cell. So nucleic acid, fourth step enagutte, biosynthesis. The nucleic acid has to be replicated. It will replicate. After it is replicated, then our last step, what is happening, assembly and release. So protein, nucleic acids are produced, proteins are synthesized. So these are assembled into a virus. Viruses assemble agutte matte ee viruses particles na release agutte. How? By killing the cell. By the lysis of the cell, these viruses are released. So these are the 5 steps in the replication process of a virus. Now, when we bacteria cell can undergo one of 2 types of infections by viruses, called as the lytic infection and lysogenic infections. ಈ ಲೈಟಿಕ್ ಇನ್ಫೆಕ್ಷನ್ ಅಲ್ಲಿ ಏನಾಗುತ್ತೆ, ದ ವೈರಸ್ ಸೇಮ್ ರೆಪ್ಲಿಕೇಶನ್ ಹೇಗಾಗುತ್ತೆ, ದ ವೈರಸ್ ಬಂದ್ಬಿಟ್ಟು ಹೋಸ್ಟ್ ಸೆಲ್ಗೆ ಅಟ್ಯಾಚ್ ಆಗುತ್ತೆ. ಹೋಸ್ಟ್ ಸೆಲ್ಗೆ ಅಟ್ಯಾಚ್ ಆಗ್ಬಿಟ್ಟು ಆ DNA ವೈರಸ್ DNA ಏನಿದೆ, ಜಿನೋಮ್ ಇದೆ, ಅದು ಹೋಸ್ಟ್ ಸೆಲ್ ಒಳಗೆ ಬರುತ್ತೆ. ಅದು ಬಂದ್ಬಿಟ್ಟು ಅಲ್ಲಿ ಒಂದು ಸರ್ಕ್ಯುಲರ್ DNA ಫಾರ್ಮ್ ಆಗುತ್ತೆ. After it is formed, depending on the conditions, so it will determine whether it has to go to the lytic cycle or the lysogenic cycle. So lytic cycle ಏನಾಗುತ್ತೆ, the cell, so the genome is there, the genome will take up the host machinery and it starts replicating its own genome. Viruses nucleic acid replicate agutte, idralli replicate ಆದ್ಮೇಲೆ same assembly ಆಗುತ್ತೆ, virus particles are getting assembled and then the cell lyses and it releases the virus particles. So this is the lytic cycle. Lytic cycle andre here the host cell is killed, the host cell is lysed and the viral particles are produced. So the lytic cycle. Other type of infection is lysogenic. ಈ lysogenic cycle ಅಲ್ಲಿ ಏನಾಗುತ್ತೆ, the same process. First attachment ಆಗುತ್ತೆ, first the viruses comes and attach to the bacteria, the host cell, that is the bacteria here. So attach ಆದ್ಮೇಲೆ ನ್ಯೂಕ್ಲಿಕ್ ಆಸಿಡ್ ಏನಿದೆ ವೈರಲ್ದು ಅದು ಒಳಗೆ ಬಂದ್ಬಿಟ್ಟು ಅದೇನಾಗುತ್ತೆ, ಡಿಪೆಂಡ್ ಆನ್ ದ ಕಂಡೀಷನ್ಸ್. ಇಫ್ ದ ನಂಬರ್ ಆಫ್ ವೈರಸ್ ಪಾರ್ಟಿಕಲ್ಸ್ ಜಾಸ್ತಿ ಇದ್ರೆ, ಸೊ ಇಟ್ ಏನಾಗುತ್ತೆ, ಅದು ಹೋಗ್ಬಿಟ್ಟು ಬ್ಯಾಕ್ಟೀರಿಯಾ DNA ಇಂಟಿಗ್ರೇಟ್ ಆಗುತ್ತೆ. ಇಟ್ ವಿಲ್ ಗೆಟ್ ಇಂಟಿಗ್ರೇಟೆಡ್ ವಿತ್ ದ ಬ್ಯಾಕ್ಟೀರಿಯಲ್ DNA. ಸೊ ಒನ್ಸ್ ಇಟ್ ಈಸ್ ಇಂಟಿಗ್ರೇಟೆಡ್, ಸೊ ದ ಬ್ಯಾಕ್ಟೀರಿಯಾಗೆ ಏನು ಎಫೆಕ್ಟ್ ಆಗಲ್ಲ, ಅದು ನಾರ್ಮಲಿ ಮಲ್ಟಿಪ್ಲೈ ಆಗ್ತಾ ಇರುತ್ತೆ. ಇಟ್ ವಿಲ್ ಸ್ಟಾರ್ಟ್ ಮಲ್ಟಿಪ್ಲೈಂಗ್. ಸೊ ಈಚ್ progeny has a viral DNA here. Each progeny has a viral DNA, so that it is just present, it's just passed on from generation to generation. But when the condition, when the cell is under stress, when it is stressed, what happens, this viral genome, it gets separated from the bacterial DNA. So when the genome is integrated with the bacterial DNA, ಅದನ್ನ ನಾವು prophage ಅಂತ ಹೇಳ್ತೀವಿ. ಸೊ ಈ ಕಂಡೀಷನ್ ಸ್ಟ್ರೆಸ್ ಇದ್ರೆ ಏನಾಗುತ್ತೆ, ಆ ಪ್ರೋಫೇಜ್ ಬ್ಯಾಕ್ಟೀರಿಯಲ್ ಡಿ ಎನ್ ಎ ಇಂದ ಸೆಪರೇಟ್ ಆಗಿಬಿಟ್ಟು ಇಟ್ ಸ್ಟಾರ್ಟ್ಸ್ ದ ಲೈಟಿಕ್ ಸೈಕಲ್. ಲೈಟಿಕ್ ಸೈಕಲ್ ಅಂದ್ರೆ ಅಲ್ಲಿ ಹೋಗ್ಬಿಟ್ಟು ಅಸೆಂಬ್ಲಿ ಆಗಿಬಿಟ್ಟು ಇಟ್ ಸ್ಟಾರ್ಟ್ಸ್ ಬಯೋಸಿಂಥಸೈಸಿಂಗ್ ಇಟ್ಸ್ DNA, nucleic acid is replicated, proteins are synthesized, then these things are assembled into a viral particle and the cell is lysed so that these particles are released. So under viral replication, we can have two types of cycles. When the virus infects a bacterial cell, the bacterial cell can undergo two types of cycles, that is lytic cycle and the lysogenic cycle. So in lytic cycle, the host cell is died, it is killed so that the viral particles are released. Whereas in lysogenic cycle, the viral DNA gets integrated in the bacterial DNA. It forms a prophage, and this prophage will be carried out to a generation until there is any stress. When there is a stress to the cell, what happens? The viral DNA gets separated. It gets separated from the bacterial DNA, and then it undergoes the lytic cycle. So this is about the lytic and the lysogenic cycle. So the bacterial cell undergoes this. So next one we took bacteria ಬಗ್ಗೆ ಮಾತಾಡೋಣ. So bacteria, the milestone events in the history of microbiology. It start ಆಗಿರೋದು 1676 to 2004. In 1676 ಅಲ್ಲಿ Anton Leeuwenhoek he observed small animalcules and discovered bacteria in 1684. Who discovered bacteria? It was Anton van Leeuwenhoek who discovered bacteria in the year 1684. Now bacteria ಅಂತ ಹೇಳಿದ್ರೆ these are prokaryotic microorganisms. Prokaryote ಯಾಕೆ ಹೇಳ್ತೀವಿ ಅಂದ್ರೆ ಇದಕ್ಕೆ ಟ್ರೂ ನ್ಯೂಕ್ಲಿಯಸ್ ಇಲ್ಲ, ದೇ ಡೋಂಟ್ ಹ್ಯಾವ್ ಅ ಟ್ರೂ ನ್ಯೂಕ್ಲಿಯಸ್ ಅಂಡ್ ದೇ ಆರ್ ಯೂನಿಸೆಲ್ಯುಲರ್, ದೇ ಡು ನಾಟ್ ಎಕ್ಸಿಬಿಟ್ ಟ್ರೂ ಬ್ರಾಂಚಿಂಗ್. Bacteria are prokaryotic microorganisms that do not contain a true nucleus. Bacteria prokaryotic microorganism ಯಾಕೆ? Because ಅದರಲ್ಲಿ ಟ್ರೂ ನ್ಯೂಕ್ಲಿಯಸ್ ಇಲ್ಲ. ನ್ಯೂಕ್ಲಿಯಸ್ is not covered by a nuclear membrane. So it does not contain a true nucleus and are unicellular, single cellular. Unicellular under single celled organisms and they don't exhibit true branching. ನಾನೀಗ prokaryote ಮತ್ತೆ eukaryote ಅಂತ ಹೇಳಿದೆ. So there is a slide here I want to show here. So in prokaryote, see there is difference here. Difference ಕಾಣಿಸುತ್ತೆ ಕ್ಲಿಯರ್ ಆಗಿ. ಒಂದು prokaryotic cell, ಇನ್ನೊಂದು eukaryotic cell. Prokaryotic cell ಅಲ್ಲಿ you can see center region ಅಲ್ಲಿ nucleus ಇದೆ. So that area, that particular area where the nucleus is there, it is called as nucleoid. The central region where nucleus is there, it is nucleoid region. And in nucleoid region, see, ಹೊರಗೆ ಏನಿರುತ್ತೆ, cell wall ಇರುತ್ತೆ, ಹೊರಗೆ cell wall ಇದೆ. So inside the cell wall you have the cytoplasm and other organelles are there, ribosomes, other things are there. And but this nucleoid, this is the major difference between the prokaryote and eukaryote. In prokaryote, there is no true nucleus. The nucleus is not covered by a nuclear membrane. It is just present as such in a central area. That area, our area na ಏನು ಹೇಳ್ತೀವಿ ನಾವು ನ್ಯೂಕ್ಲಿಯಾಯ್ಡ್ ಅಂತ ಹೇಳ್ತೀವಿ. ಸೊ ಅದು ನ್ಯೂಕ್ಲಿಯಾಯ್ಡ್ ಇದೆ. And coming to eukaryotes, eukaryotes ಅಂದ್ರೆ they are already well defined nucleus ಇರುತ್ತೆ. Well defined nucleus is there. So the nucleus, it is surrounded by a nuclear membrane. ಮತ್ತೆ membrane bound organelles ಇರುತ್ತೆ. Cytoplasmic membranes are there, endoplasmic reticulum, Golgi complex, mitochondria. These are all membrane bound organelles which are present in eukaryotes. So major difference, prokaryotes ಮತ್ತು eukaryotes major difference ಏನಿದೆ? The nucleus. In prokaryotes, there is no true nucleus. They do not contain a true nucleus. That is, nucleus is present as such in the center, called as a nucleoid, whereas it is not surrounded by nuclear membrane. But eukaryotes ಏನಾಗುತ್ತೆ? The nucleus is surrounded by a nuclear membrane. A proper membrane is there, a nuclear membrane. That is the difference here between the prokaryote and eukaryote. So bacteria idu prokaryote. Bacteria are prokaryotic microorganisms and they are unicellular, single celled organisms idu. Next ಬಂದ್ಬಿಟ್ಟು classification. ಇದನ್ನ ಹೇಗೆ ಕ್ಲಾಸಿಫೈ ಮಾಡ್ತೀವಿ ನಾವು? So bacteria, how can we classify? Generally based on shape, bacteria shapes ಏನಿದೆ ಅದ್ರ ಪ್ರಕಾರ ನಾವು ಬ್ಯಾಕ್ಟೀರಿಯಾಸ್ನ ಕ್ಲಾಸಿಫೈ ಮಾಡ್ಬೋದು. So based on shape in ಬಗ್ಗೆ ನಾವು ಕ್ಲಾಸಿಫೈ ಮಾಡಬೇಕಂದ್ರೆ ಫಸ್ಟ್ ವಿಲ್ ಬಿ ಆರ್ ಕಾಕೈ. ಕಾಕೈ ಮೀನ್ಸ್ ದ ಬ್ಯಾಕ್ಟೀರಿಯಾಸ್ ಆರ್ ಸ್ಪೆರಿಕಲ್. ಬ್ಯಾಕ್ಟೀರಿಯಾಸ್ ಅಲ್ಲಿ ಸ್ಪೆರಿಕಲ್ ಶೇಪ್ ಅಲ್ಲಿ ಇರುತ್ತೆ, ಸರ್ಕ್ಯುಲರ್ ಶೇಪ್ ಅಲ್ಲಿ ಇರುತ್ತೆ. ಅದನ್ನ ಸರ್ಕ್ಯುಲರ್ ಶೇಪ್ ಅಲ್ಲಿ ಇರೋ ಬ್ಯಾಕ್ಟೀರಿಯಾನ ನಾವು ಕಾಕೈ ಅಂತ ಹೇಳ್ತೀವಿ. ಸೆಕೆಂಡ್ ಬಂದ್ಬಿಟ್ಟು ಬ್ಯಾಸಿಲೈ. ಬ್ಯಾಸಿಲೈ ಬಂದ್ಬಿಟ್ಟು ರಾಡ್ ಶೇಪ್ ಅಲ್ಲಿ ಇರುತ್ತೆ. ರಾಡ್ ಶೇಪ್ ಬ್ಯಾಕ್ಟೀರಿಯಾ ಬಂದ್ಬಿಟ್ಟು ನಮ್ದು ಬ್ಯಾಸಿಲೈ. So next type of bacteria, next class of bacteria is Vibrio, a comma shaped. Now comma Vibrio cholera ಇದೆ. So the bacteria shape ಏನಿದೆ comma shaped ಇರುತ್ತೆ, ಅದಕ್ಕೆ ನಾವು ಅದನ್ನ Vibrio ಅಂತ ಹೇಳ್ತೀವಿ. ನೆಕ್ಸ್ಟ್ ಬಂದ್ಬಿಟ್ಟು ಲಾಸ್ಟ್ ಬಂದ್ಬಿಟ್ಟು ಸ್ಪೈರಲಾ. ಸ್ಪೈರಲಾ ಅಂದ್ರೆ here the bacterial shape will be in the form of spirals. ಸ್ಪೈರಲ್ ಶೇಪ್ ಅಲ್ಲಿ ಇರುತ್ತೆ, ಅದನ್ನ ನಾವು ಸ್ಪೈರಲ್ ಅಂತ ಹೇಳ್ತೀವಿ, ಸ್ಪೈರಲಾ. ಸೊ ಬ್ಯಾಕ್ಟೀರಿಯಾಸ್ ಬೇಸ್ಡ್ ಆನ್ ಶೇಪ್ ಅದು ಹೇಗಿದೆ ಆ ಶೇಪ್ ಇಂದ ಮೂಲಕ ನಾವು ಇದನ್ನ 4 ಕ್ಲಾಸಸ್ ಅಲ್ಲಿ ಕ್ಲಾಸಿಫೈ ಮಾಡ್ತೀವಿ. ಒನ್ ಇಸ್ ಕಾಕೈ, ಸೆಕೆಂಡ್ ಇಸ್ ಬ್ಯಾಸಿಲೈ, ಥರ್ಡ್ ಇಸ್ ವಿಬ್ರಿಯೋ ಅಂಡ್ ಸ್ಪೈರಲಾ. ಕಾಕೈ ಮೀನ್ಸ್ ಇಟ್ ಈಸ್ ಸರ್ಕ್ಯುಲರ್ ಶೇಪ್ ಅಲ್ಲಿ ಇರೋ ಬ್ಯಾಕ್ಟೀರಿಯಾನ ನಾವು ಕಾಕೈ ಅಂತ ಹೇಳ್ತೀವಿ. ರಾಡ್ ಶೇಪ್ ಅಲ್ಲಿ ಇರೋ ಬ್ಯಾಕ್ಟೀರಿಯಾನ ಬ್ಯಾಸಿಲೈ ಅಂತ ಹೇಳ್ತೀವಿ. ಮತ್ತೆ ಕಾಮಾ ಶೇಪ್ ಇರೋ ಬ್ಯಾಕ್ಟೀರಿಯಾನ ನಾವು ವಿಬ್ರಿಯೋ ಅಂತ ಹೇಳ್ತೀವಿ. ಲಾಸ್ಟ್ ಬಂದ್ಬಿಟ್ಟು ಸ್ಪೈರಲ್ ಶೇಪ್ ಇದ್ರೆ ಅದನ್ನ ಸ್ಪೈರಿಲಾ. ಇದು ಒಂದ್ ಪ್ರಕಾರ ನಾವು ಕ್ಲಾಸಿಫೈ ಮಾಡ್ಬೋದು. ಇನ್ನೊಂದು ಪ್ರಕಾರ ಅಂದ್ರೆ ಬೇಸ್ಡ್ ಆನ್ ದ ಅರೇಂಜ್ಮೆಂಟ್ ಆಫ್ ದ ಸೆಲ್ಸ್, ಬ್ಯಾಕ್ಟೀರಿಯಾ ಆರ್ ಕ್ಲಾಸಿಫೈಡ್ ಆಸ್ ಡಿಪ್ಲೋಕಾಕೈ, ಸ್ಟ್ರೆಪ್ಟೋಕಾಕೈ, ಟೆಟ್ರಾಡ್ಸ್ ಅಂಡ್ ಕ್ಯೂಬಿಕಲ್ ಪ್ಯಾಕೆಟ್ಸ್. ಬೇಸ್ಡ್ ಆನ್ ಅರೇಂಜ್ಮೆಂಟ್ ಆಗಿ ನಾವು ಸೆಲ್ಸ್ ಬ್ಯಾಕ್ಟೀರಿಯಾಸ್ ನ ಹೇಗೆ ಕ್ಲಾಸಿಫೈ ಮಾಡ್ತೀವಿ ಅದನ್ನ ಮತ್ತೆ 4 ಕ್ಲಾಸಸ್ ಎಲ್ಲ ಕ್ಲಾಸಿಫೈ ಮಾಡ್ತೀವಿ. ದ ಫಸ್ಟ್ ಕ್ಲಾಸ್ ಬಂದ್ಬಿಟ್ಟು ಡಿಪ್ಲೋಕಾಕೈ. ಡಿಪ್ಲೋ ಮೀನ್ಸ್ ನೇಮ್ಸ್ ಹೇಳುತ್ತೆ 2 cells together. 2 cocci when it comes together it is diplococci. Second is streptococci, a group of cells. I'll show you the slide. Streptococci. Then tetrads. It is arranged in 4 cells. Tetrads. But the cubical packets. Last thing. See here, as you can see here in the picture, Diplococci, Streptococcus pneumoniae. So 2 cocci cells are together here. That is your Diplococci. Then Streptococci, it is the cells are arranged in a filament form. So that is Streptococci. That is example banbittu Streptococcus pyogenes. Example kodabodu navu idakke. Matte tetrad ide. Sarsinia, again a group of cells. Arrangement. Group of cells idre adu Sarsinia irutte. And Staphylococcus, again a group of cells together. It is Staphylococcus. Then rod shape, I told you, rod shape andre bacilli. So bacilli, again if a chain of bacillus iridre adanna bacillus anthracis ಹೇಳ್ತೀವಿ. ಆ bacillus ಅಲ್ಲಿ flagella present ಇದ್ರೆ flagellated rods ಅಂತ ಹೇಳ್ತೀವಿ. Example ಬಂದ್ಬಿಟ್ಟು Salmonella typhi, which causes your typhoid. Then spirals, vibrio shaped. Vibrios means comma shaped irutte, adanna ನಾವು vibrio cholerae ಅಂತ ಹೇಳ್ತೀವಿ. And last spiral shape, that is Helicobacter pylori. This is the examples for spirilla bacteria. So based on shape, bacteria are classified into 4 main groups. Based on shape, bacteria ನಾವು ಹೇಗೆ ಕ್ಲಾಸಿಫೈ ಮಾಡ್ತೀವಿ 4 groups ಅಲ್ಲಿ ಕ್ಲಾಸಿಫೈ ಮಾಡ್ತೀವಿ. ಒಂದು ಕಾಕೈ, ಸೆಕೆಂಡ್ ಇಸ್ ಬ್ಯಾಸಿಲೈ. ಕಾಕೈ ಅಂದ್ರೆ ಸರ್ಕ್ಯುಲರ್ ಶೇಪ್, ಬ್ಯಾಸಿಲೈ ಅಂದ್ರೆ ರಾಡ್ ಶೇಪ್, ವಿಬ್ರಿಯೋ ಅಂದ್ರೆ ಕಾಮಾ ಶೇಪ್, and spiral shape ಅಲ್ಲಿ ಇರುತ್ತೆ bacteria. So that is how we classify the bacteria. Next, coming to the bacteria cell ಅದರ ರಚನೆ ಹೇಗಿದೆ ಅಂತ ನೋಡೋಣ. ಫಸ್ಟ್ ಏನಿದೆ ಬ್ಯಾಕ್ಟೀರಿಯಾ ಸೆಲ್, ಇಟ್ ಇಸ್ ಕವರ್ಡ್ ಬೈ ಸೆಲ್ ವಾಲ್. ಔಟರ್ ಮೋಸ್ಟ್ ಲೇಯರ್, ಹೊರಗಿನ ಲೇಯರ್ ಅನ್ನ ಏನ್ ಹೇಳ್ತೀವಿ ಸೆಲ್ ವಾಲ್ ಅಂತ ಹೇಳ್ತೀವಿ. ಯು ಕ್ಯಾನ್ ಸಿ ಇನ್ ದ ಪಿಕ್ಚರ್ ಹಿಯರ್ ಇನ್ ದ ಸ್ಲೈಡ್. ಸೊ ಸೆಲ್ ವಾಲ್ ಇಸ್ ದೇರ್. ಸೊ ಇನ್ನರ್ ಸೆಲ್ ವಾಲ್ ಒಳಗೆ ಏನಿರುತ್ತೆ ಪ್ರೋಟೋಪ್ಲಾಸಮ್ ಇದೆ. ಸೆಲ್ ವಾಲ್ ಒಳಗೆ ಏನಿದೆ ಇನ್ನರ್ ಪ್ರೋಟೋಪ್ಲಾಸಮ್ ಇದೆ. ಅಂಡ್ ಆ ಪ್ರೋಟೋಪ್ಲಾಸಮ್ ಒಳಗೆ ಬೇರೆ ಬೇರೆ ಕಾಂಪೊನೆಂಟ್ಸ್ ಪ್ರೆಸೆಂಟ್ ಇರುತ್ತೆ. ಜೈಸೆ ಪ್ಲಾಸ್ಮಿಡ್ಸ್ ಅಂತ ಇದೆ. ಪ್ಲಾಸ್ಮಿಡ್ಸ್ ಏನು? ಪ್ಲಾಸ್ಮಿಡ್ಸ್ ಆರ್ ನಥಿಂಗ್ ಬಟ್ extra small circular DNA. ಈಗ bacterial cell ಅಲ್ಲಿ nucleoid region where DNA is present. So in that bacteria we have a small circular DNA which can replicate independently. So that is called plasmids. Then we have ribosomes, microcompartments. Microcompartments ಅಂದ್ರೆ membrane bound structures. ಅದರಲ್ಲಿ protein shell ಇರುತ್ತೆ, ಅದರ ಒಳಗೆ some enzymes ಮತ್ತೆ proteins enclose ಆಗಿರುತ್ತೆ. Gas vesicles are there, ಮತ್ತೆ magnetosomes irutte. So this is the shape of bacteria. Bacteria comes under prokaryotes. Prokaryotes andre no proper nucleus, no true nucleus is present. So ಶೇಪ್ ರಚನೆ ಬಗ್ಗೆ ಮಾತಾಡಿದ್ರೆ ಹೊರಗಿನ ಲೇಯರ್ ನಾವು ಸೆಲ್ ವಾಲ್ ಹೇಳ್ತೀವಿ. ಬ್ಯಾಕ್ಟೀರಿಯಾ ಹ್ಯಾಸ್ ಸೆಲ್ ವಾಲ್. ಸೊ ಇನ್ನರ್ ಟು ದ ಸೆಲ್ ವಾಲ್ ಇಸ್ ದ protoplasm and then other components irutte like plasmids. I told you plasmids andre eno. Plasmids andre small circular extra small chromosomal DNA which can replicate itself independent agi replicate agutte adu. So then ribosomes irutte, micro compartments irutte, gas vesicles and then magnetosomes. Ela components irutte bacteria cell alli. So this is the bacteria cell structure, simple structure. Now, bacteria, they have a noteworthy impact. It is having a very good impact on human life because of their beneficial as well as harmful activities. It is beneficial as well as harmful. So it is now possible for us to manipulate microbes for the benefit of mankind by using the latest advances in the biological sciences and technology. So here, bacteria, they are playing a major role. It is playing a major role in food industry, in medicine, environmental management, and agriculture. So bacteria are playing a very good role here. So that is, it has a vast importance. Now coming to the reproduction here, bacteria reproduction. So as I told you earlier, in bacteria, the genetic material, it is organized as a continuous circular strand of DNA. It is organized as a continuous circular strand of DNA, and this is called present in the area called nuclide. In the nuclear region, DNA is present. Now, these cells, they have extra chromosomal circular DNA strand that is called as plasmids. These plasmids can replicate independently. Plasmids, they can replicate independently. So bacteria, when we talk about reproduction, bacteria, they can exhibit, both asexual and sexual reproduction. Asexual reproduction means only one cell is involved here. So one cell will divide to give rise to daughter cells. That is asexual reproduction. And in sexual reproduction, two cells are involved here. So that is sexual reproduction. So this bacteria, it can exhibit both asexual as well as sexual reproduction. So first we'll see how asexual reproduction is taking place. So as you can see here in the slide, asexual reproduction is mainly through binary fission. Asexual reproduction takes place mainly by binary fission. Then endospore formation also takes place, exospores, or by cyst formation. So first we'll discuss about binary fission. So in binary fission, you have one single bacteria cell ಇದೆ. So binary one cell divides to give rise to two daughter cells. So one cell we have, that is bacterial cell is there. So in this, which is called as nothing but the parent cell. So this parent cell, the bacterial parent cell, it will duplicate. The DNA of the parent cell duplicates into two daughter cells. So first what is happening? First step ಏನಾಗುತ್ತೆ? DNA duplicate ಆಗುತ್ತೆ. Parent cell ಅಲ್ಲಿ DNA duplicate ಆಗುತ್ತೆ. ಆಮೇಲೆ cytoplasm division ಆಗುತ್ತೆ. So once DNA is duplicated, the cytoplasm will divide. So now ಏನು ಸಿಗುತ್ತೆ ನಮಗೆ? ಎರಡು ಸೆಲ್ ಸಿಗುತ್ತೆ. ಸೊ ಅದು ಬೈನರಿ ಫಿಷನ್. ಬೈನರಿ ಫಿಷನ್ ಅಂದ್ರೆ ಒನ್ ಪೇರೆಂಟ್ ಸೆಲ್ ಡಿವೈಡ್ ಆಗಿಬಿಟ್ಟು ಟೂ ಡಾಟರ್ ಸೆಲ್ ಸಿಗುತ್ತೆ ನಮಗೆ. ಸೊ ದಟ್ ಇಸ್ ಬೈನರಿ ಫಿಷನ್. Once again, here binary fission means one parent cell, that is one parent cell divides to give rise to two daughter cells. That is binary fission. So binary fission is there. Then what happens? There is another thing of reproduction, that is a dormant, heat resistant and non-growing cell develops during endospore formation. Endospore, when the conditions are not favorable. When the conditions are not favorable, that what happens? The cell multiplies and it forms endospores. So we'll see how this endospore formation takes place. So the first step here is you have your bacteria cell. So in bacteria cell the DNA ಇದೆ ತಾನೆ, ಆ DNA replicate ಆಗುತ್ತೆ. So the DNA is replicated. The first step in the endospore formation, DNA is replicated. It is replicated now. Once DNA is replicated, Second step, what happens? Cellular division takes place. Cellular division of the cytoplasmic membrane. DNA ಇದೆ, DNA replicate ಆಗಿದೆ, ಮತ್ತೆ cytoplasm cellular division takes place. Cellular division by cytoplasmic membrane takes place. So now third step is pre-spore formation. Pre-spore formation begin ಆಗುತ್ತೆ. So once DNA is there, the cytoplasmic division has taken place. So ಆ ಏರಿಯಾದಲ್ಲಿ pre-spore formation takes place. A septum is formed there. So pre-spore formation takes place. Then mother cell ಇದೆ, mother cell ಅಲ್ಲಿ nucleate DNA duplicate ಆಗಿದೆ, DNA is doubled, DNA is replicated. Then cytoplasmic division takes place. That is a cellular division of the cytoplasmic membrane takes place. Now a septum, a wall is formed, a pre-spore formation begin ಆಗುತ್ತೆ. ಅದು ಥರ್ಡ್ ಸ್ಟೆಪ್, ಪ್ರೀಸ್ಪೋರ್ ಫಾರ್ಮೇಷನ್. ಅದಾದ್ಮೇಲೆ ಏನಾಗುತ್ತೆ? ಕಾರ್ಟೆಕ್ಸ್ ಫಾರ್ಮೇಷನ್ ಸ್ಟಾರ್ಟ್ ಆಗುತ್ತೆ. ಪ್ರೀಸ್ಫಾರ್ಮ್ ಆದ್ಮೇಲೆ ದಟ್ ಪರ್ಟಿಕ್ಯುಲರ್ ಸೆಲ್ ಹ್ಯಾಸ್ ಕಾರ್ಟೆಕ್ಸ್ ಫಾರ್ಮೇಷನ್. ಕಾರ್ಟೆಕ್ಸ್ ವಿಲ್ ಫಾರ್ಮ್ ಹಿಯರ್. Then the next step is spore coat formation begins. Once the septum and the spore is formed, then the cortex is formed. Then what happens? Slowly spore coat formation takes place. A spore coat is formed. ಅದಾದ್ಮೇಲೆ ಆ ಸ್ಪೋರ್ ಏನಾಗುತ್ತೆ? ಅದು ಮೆಚ್ಯೂರ್ ಆಗುತ್ತೆ. ಮೆಚುರೇಷನ್ begins here, and exosporium, another layer is formed there called as exosporium. So now maturation is taking place. It has matured now. And then the mother cell ಏನಿದೆ ಅದು ಆ ಸೆಲ್ ನ ಸ್ಪೋರ್ ನ ರಿಲೀಸ್ ಮಾಡುತ್ತೆ, so that your free spore is produced. So this is how the spore formation is taking place. So once again I'll repeat here, when there is a dormant condition, when there is heat resistant, when there is conditions are not favorable, then spore formation takes place. Endospore formation. Endo means the spore is formed inside the cell. Endospore formation takes place. So how it is taking place, how the endospore is formed? So we have multiple steps. First step ಏನಾಗುತ್ತೆ? The DNA is replicated. First step ಅಲ್ಲಿ the DNA, the nucleic acid, the genome is replicated. So once the DNA is replicated, then second step ಏನಾಗುತ್ತೆ? The cellular division of the cytoplasmic membrane takes place. Cellular division, cytoplasmic membrane division ಆಗುತ್ತೆ. ಅದಾದ್ಮೇಲೆ pre-spore formation start ಆಗುತ್ತೆ. Once cellular division of the cytoplasm ಆದ್ಮೇಲೆ pre-spore formation start ಆಗುತ್ತೆ. It begins. Pre-spore formation begins. So here once this pre-spore formation is begins, so slowly means one septum start ಆಗುತ್ತೆ. ಆಮೇಲೆ cortex formation start ಆಗುತ್ತೆ. Cortex formation takes place. That is the fourth step here, cortex formation. Once cortex is formed, then what happens? Spore coat formation takes place. Cortex is there, cortex is formed now. So around this cortex, what is happening? Seed coat formation begins. So spore coat formation begins. So once the spore coat is formed, then another layer, it undergoes maturation. ಏನಾಗುತ್ತೆ? Exosporium formation takes place ಆಗುತ್ತೆ. So once maturation begins, our exosporium is formed. So now endospore form ಆಗಿದೆ. So ಈಗ ಅದು ರಿಲೀಸ್ ಆಗಬೇಕು. When the conditions are favorable, what happens? These exospores, the mother cell releases your mature spore. So that free spore, again, when the conditions are favorable, it will plasm division, the division of the cytoplasmic membrane, then spore formation begins, then maturation, then cortex formation takes place, then spore coat formation begins, and then maturation begins. That is exosporium formation takes place. Finally, when this is formed, the mother cell will release our mature spore, so that free spore is released. So next is exospores. See, exospores, Andre, the spores are produced outside the parent cell. That is the difference between endospore and exospore. Endospore ಮತ್ತು exospore difference ಏನಿದೆ? Endospore ಅಂದ್ರೆ endo ಅಂದ್ರೆ ಒಳಗೆ, ಪೇರೆಂಟ್ ಸೆಲ್ ಒಳಗೆ जो ಪ್ರೊಡ್ಯೂಸ್ ಆಗುತ್ತೆ ಅದನ್ನ ಎಂಡೋಸ್ಪೋರ್ ಹೇಳ್ತೀವಿ. Exospore which is present outside the parent cell. So exospores and cysts, they are desiccation resistant resting cells. Usually these are formed by some methanotropes. These exospores and cysts, they are formed from methanotropes. So you can see here in the slide, the differences have shown. Endospore means this is present within the cell. Exospore, the spore which is present outside the cell. So endospore ಮತ್ತೆ exospore. Next is sexual reproduction. ಈಗ ನಾವೇನ್ ಮಾತಾಡಿದ್ವಿ asexual reproduction ಬಗ್ಗೆ ಮಾತಾಡಿದ್ವಿ. ಈಗ sexual reproduction ಬಗ್ಗೆ ಮಾತಾಡೋಣ. So sexual reproduction. So sexual reproduction is not an obligatory step. It is not compulsory important in the life of a bacterium. So sexual reproduction in bacteria, it is not an obligatory step in the life of a bacterium. So this sexual reproduction is nothing but it is the exchange of the genetic material. The mechanism of exchange of genetic material in bacteria, this can happen by means of three ways. The three ways of sexual reproduction ಆಗುತ್ತೆ. ಒಂದು ಬಂದ್ಬಿಟ್ಟು conjugation, second is transformation, and third is transduction. Now first we'll see about conjugation. So conjugation ಏನಾಗುತ್ತೆ, here genetic exchange ಆಗಿದೆ between two bacterial cells. Two bacterial cells ಅಲ್ಲಿ genetic exchange ಆಗಿದೆ through direct physical contact. Through direct physical contact, the genetic material is exchanged between two bacterial cells. So principally here we have plasmids or the F factor, that is fertility factor. So this is how conjugation takes place. Let me explain you the steps here. So two cells are there. The first, the cell, the donor cell ಬಂದ್ಬಿಟ್ಟು ಅದು F plus fertility factor present ಇರುತ್ತೆ. fertility factor present ಇರೋ ಸೆಲ್ ನ ನಾವು F plus cell ಅಂತ ಹೇಳ್ತೀವಿ. ಇನ್ನೊಂದು ಬಂದ್ಬಿಟ್ಟು F minus cell. F minus cell ಅಂದ್ರೆ ಆ ಸೆಲ್ ನಲ್ಲಿ fertility factor ಇಲ್ಲ. So donor cell has F plus cell. It is also called as F plus cell. The recipient cell, it is called as F minus cell. So first step ಏನಾಗುತ್ತೆ ಇದರಲ್ಲಿ, first the donor cell attaches to the recipient cell. Donor cell attach ಆಗುತ್ತೆ recipient cell with its pilus. So pilus ಏನಾಗುತ್ತೆ, pilus draw ಮಾಡುತ್ತೆ, it draws the cell together. The pilus will pull the cells together. So they come into physical contact here. So the first cell, second step, first cell, the donor cell attaches to a recipient cell with its pilus. So ಈ pilus ಏನ್ ಮಾಡುತ್ತೆ, pilus ಎರಡು cell ನ draw ಮಾಡುತ್ತೆ, it draws the cell together. Next is the cells contact one another. There is a physical contact between the two cells. In conjugation ಏನಾಗುತ್ತೆ, this involves genetic exchange between two bacterial cells through physical contact. To direct physical contact is there here. So when the donor cell attaches to a recipient cell through the pilus draws the cell together. So the cells contact one another. So when the cells contact one another, the F plus cell, that is a fertility factor ಇರೋ ಸೆಲ್ ಏನಾಗುತ್ತೆ, ಅದರಿಂದ one strand ಬಂದ್ಬಿಟ್ಟು ಈ ಬೇರೆ ಸೆಲ್ಗೆ ಬರುತ್ತೆ. One strand of the plasmid DNA transfers to the recipient. From the F plus cell in the one strand of the DNA is transferred to the recipient cell. It is transferred. Next step ಏನಾಗುತ್ತೆ, the recipient cell synthesizes a complementary strand. ಆ strand ಏನ್ ಪಾಸ್ ಆಗಿದೆ ಅದಕ್ಕೆ complementary strand synthesize ಮಾಡುತ್ತೆ. So that the F minus cell becomes a F plus cell. Okay, and the donor cell allೂ ಕೂಡ ಒಂದೇ ಒಂದು strand ಇರೋದು, even that also synthesizes a complementary strand. So therefore it is restoring its complete plasmid. So this is how the exchange is taking place between the two bacterial cells. So in conjugation ಏನಾಗುತ್ತೆ, two bacterial cells ಅಲ್ಲಿ direct physical contact ಇದೆ. Through physical contact the genetic material is exchanged. So first two cells, donor cell ಮತ್ತೆ recipient cell. Donor cell ಬಂದ್ಬಿಟ್ಟು F plus cell, we call that as F plus because it is having a plasmid, a fertility factor is present. Recipient cell, F minus cell, it is not having a fertility factor or a plasmid. So this donor cell ಬಂದ್ಬಿಟ್ಟು recipient cell ಗೆ attach ಆಗುತ್ತೆ through pilus. So pilus ಏನ್ ಮಾಡುತ್ತೆ, ಆ ಎರಡು cell close ಮಾಡುತ್ತೆ, it brings the cells together. Then a contact is formed, a tube, a conjugation tube is formed. The two cells are in contact now. So when the two cells are in contact now, the donor cell in the one strand, F plus factor ಇದೆ, so that one strand it moves to the other cell, that is the recipient cell. Through the conjugation tube it is moving. So one strand from the F plus cell it moves to the recipient cell. So once the one strand has come to the recipient cell, it will be complete synthesizes a complementary strand. It synthesizes a complementary strand to become F plus. And F plus cell, donor cell F plus one strand, even that also will synthesize a complementary strand so that it becomes a complete plasmid. So this is conjugation. Conjugation means there is contact, there is a physical contact between two bacterial cells, and the exchange involves genetic exchange is between two bacterial cells through physical contact. Basically we have plasmids or a F factor. So this is conjugation. Then the second type of sexual reproduction what we can see in bacteria is transformation. Now transformation means here what is happening, the DNA ಏನಿದೆ, it is transferred from where, from cells to the surrounding medium. Cells in the surrounding medium transfer ಆಗುತ್ತೆ ಅದು first DNA. ಆಮೇಲೆ from the surrounding medium it gets incorporates, ಅದು recipient cell ಅಲ್ಲಿ incorporate ಆಗುತ್ತೆ. So that is transformation. So there is no direct physical contact between the two bacterial cells. So from the donor DNA, the DNA is transformed to the surrounding medium, and from the surrounding medium, the recipient cell will take up the DNA from the surrounding medium and it incorporates it. So you can see here in the slide here. So you have DNA fragments from the donor cell, A, B, F, D, E, C, B, C. These are the DNA fragments which is present in the surrounding medium. Now you have a recipient cell. So first what happens, the recipient cell will take up the donor DNA. Recipient cell is there. So what it is doing, it is taking up the donor DNA from where, from the surrounding medium. So here there is no physical contact between two bacterial cells. So the recipient cell, it takes up the donor DNA from the surrounding medium, and this donor DNA ಏನಾಗಿದೆ, ಅದು ಬಂದ್ಬಿಟ್ಟು recipient cell ಅಲ್ಲಿ incorporate ಆಗುತ್ತೆ. It gets incorporated. The donor DNA gets incorporated into the recipient DNA. So recombination occurs. ಇದರಲ್ಲಿ ಏನಾಗುತ್ತೆ, recombination ಆಗಿದೆ because donor DNA and recipient DNA is there. So there is recombination. So then the cell ಏನಾಗುತ್ತೆ, ಅದನ್ನ ನಾವು genetically transformed cell ಅಂತ ಹೇಳ್ತೀವಿ. So in transformation ಅಲ್ಲಿ ಏನಾಗುತ್ತೆ, the DNA is transferred from cell to the surrounding medium, and then to the recipient cell incorporates it. So DNA surrounding medium ಅಲ್ಲಿ ಇರುತ್ತೆ, ಅದನ್ನ recipient cell will take up. Once it enters the recipient cell, what happens, it gets integrated, it gets incorporated into the recipient DNA. So recombination occurs now. Recombination occurs and the cell na ಏನು ಹೇಳ್ತೀವಿ ನಾವು, transformed cell ಅಂತ ಹೇಳ್ತೀವಿ, genetically transformed cell. So this transformation explain ಮಾಡಕ್ಕೆ there is one very good experiment done by Griffith. So this is commonly called as the Griffith's experiment. So here he has taken two strains of the pneumonia, that is Streptococcus pneumoniae. ಈ two strains ನಾವು ಏನು ಹೇಳಿದರೆ, one is R strain, ಇನ್ನೊಂದು ಬಂದ್ಬಿಟ್ಟು S strain. Now R strain means rough strain. That bacteria is not surrounded by a capsule. Capsule ಇಂದ surround ಆಗಿರಲ್ಲ, ಅದಕ್ಕೆ R strain ಅಂತ ಹೇಳ್ತೀವಿ, rough strain ಅಂತ ಹೇಳ್ತೀವಿ. So ಈ rough strain ನ when mice ಗೆ inject ಮಾಡಿದ್ರೆ ಏನಾಗುತ್ತೆ, mice pneumonia develop ಆಗಲ್ಲ, it will be alive. This is about the rough strain. ಇನ್ನೊಂದು strain ಇದೆ, ಅದು ಬಂದ್ಬಿಟ್ಟು smooth strain, ಅದು virulent type. Smooth strain ಅಂದ್ರೆ it is surrounded by a capsule. So ಈ strain ಅನ್ನ when mice ಗೆ inject ಮಾಡಿದ್ರೆ ಏನಾಗುತ್ತೆ, mice will develop pneumonia and it will die. So two strains are there, R strain ಮತ್ತೆ S strain. R strain is a non-virulent type. It will not kill the mice. ಇನ್ನೊಂದು ಬಂದ್ಬಿಟ್ಟು virulent form. So ಇದನ್ನ inject ಮಾಡಿದ್ರೆ ಏನಾಗುತ್ತೆ, mice will die. The mouse will die here. So then what he did another way. What he did was he took the S strain. S strain na heat kill ಮಾಡಿದ. He heat killed the S strain, which is also called as smooth strain. So heat killed smooth strain na ಯಾವಾಗ mouse ಅಲ್ಲಿ inject ಮಾಡಿದ್ರೆ ಅದೇನಾಗುತ್ತೆ, pneumonia is not developed in the mouse and mouse leaves. Okay, when he heat killed the smooth strain and then injected into the mouse did not develop pneumonia and it lived, it did not die. Then what he did in another type study, he took both the strains, rough strain togona ಮತ್ತೆ he took a rough strain as well as heat killed smooth strain. heat killed smooth strain. ಈ both the strains now mouse ಅಲ್ಲಿ inject ಮಾಡಿದ್ರು. Inject ಮಾಡಿದ್ಮೇಲೆ what happened, the mouse developed pneumonia and then it died. So here a combination, a rough strain as well as heat killed smooth strain, when this was injected into the mouse developed pneumonia and it died. So from the dead mouse, when he observed, he obtained S strain bacteria in the dead mice, because R strain, it is non-virulent type, right? It is not, it does not kill the mice. Whereas heat killed S strain, even that is also non-virulent. It does not kill the mice. So when he took a combination of both R strain and the S strain together, and when he injected it into the mouse, what happened? The mouse developed pneumonia and it died. Eventually, it died. So from the dead mice, he observed S strain bacteria. So he concluded with this experiment that there is some principle here. Something is being transferred from the heat killed S strain to the R strain, so that the R strain became S strain, virulent form. The non-virulent form, ಈಗ virulent form convert ಆಗಿದೆ. So that is the transformation experiment. So transformation, what is happening? Basically, there is no physical contact here between the two cells. There is no physical contact. So the DNA from the donor, the DNA is transferred to the surrounding medium, and from the surrounding medium, it gets incorporated into the recipient cell. And that cell will have a recombinant DNA because it is having its own DNA plus the donor DNA. So this is about the transformation. And Griffith also, he did experiment here. That is a very famous Griffith's experiment. Next is the last here about the sexual reproduction is about transduction. So in transduction, what is happening? The DNA is transferred from one bacterial cell to another. ಅದು ಹೇಗೆ ಟ್ರಾನ್ಸ್ಫರ್ ಆಗುತ್ತೆ, ಅದು ಹೇಗೆ ಹರಡುತ್ತದೆ one bacterial cell ಇಂದ ಇನ್ನೊಂದು bacterial cell ಗೆ? Through a virus, through a phage virus in the, because already when I told you the lytic cycle and the lysogenic cycle. So lytic cycle is what is that? The virus which is infecting a bacterial cell. Virus is infecting a bacterial cell, and from here, when it is infecting the bacterial cell, what happens? It penetrates, the nucleic acids of the virus is penetrated into the bacterial DNA, and sometimes it can enter the lysogenic phase. In the lysogenic phase, what happens? It gets incorporated into the bacterial genome, forms a prophage, and when it gets separated, it can carry some of the bacterial DNA. So when that will go and infect another bacteria, so that DNA from the donor bacteria, it can get into the recipient DNA. So here, this is the third type of mechanism you can see in case of bacteria, called as transduction. So in transduction, what is happening? The DNA is transferred from one bacterial cell to another through a phage virus. That is a bacteria phage. We can see here the first step, first, the phage has to infect the DNA. The phage has to come and attach to the bacterial cell. So the phage will come and it attaches to the bacterial cell. Then the phage injects its DNA. Phage ಏನು ಮಾಡುತ್ತೆ? ಅದರ DNA ನ inject ಮಾಡುತ್ತೆ, bacterial cell ಗೆ inject ಮಾಡುತ್ತೆ. Then the phage enzyme is there. It breaks down the host DNA. Phage enzyme ಏನು ಮಾಡುತ್ತೆ? Host DNA ನ break down ಮಾಡುತ್ತೆ. Then the cells will create new phages, and these phages they contain some of the host DNA. So as you can see here, first the bacteria, the phage comes and attaches to the bacteria, then it injects its DNA here. So after injecting, what happens? The phage enzyme, it breaks down the host DNA. The host DNA is broken down here, and then the cell creates new phages, and some of the phage particle can have this host DNA. They can have a bit of the host DNA. So then this infected the bacteria, the phage which is having the host DNA, now that can go and insert its donor DNA into another bacteria cell. So this infected virus or the bacteria, when it infects another bacteria cell, what happens? Again, the same process continues. So that is, the bacteriophage, it goes and attaches to the host cell. Then after attaching, what is happening? The host attaches, the phage will inject its DNA into the host. So this host injects into the host cell, and the DNA of the phage, which contains some of the bacterial DNA, so that will come, and again, what is happening? It is, again, it breaks down the host cell genome, and then again new virus particles are formed. So some of this DNA from the donor DNA is transformed into the recipient DNA, and then again the bacteria, what is here, we can say it is a transduced bacterium. So this is the other type called as transduction. So during transduction, here the DNA, again there is no physical contact here between the two bacterial cells. Two bacterial cells physical contact illa illi. Matte how the DNA is transferred is from one bacterial cell to another through a phage virus. So here the first step is the phage injects its DNA. The phage is injecting its DNA into the bacteria. Then once it injects its DNA, what happens? The phage enzyme breaks down the host DNA. The host DNA is broken down. Then in the next few two steps, what is happening? The cell will create new phages, including a phage which contains the host DNA. So new phage particles are produced, and then the host cell will lyse. The host cell is lysed to release these phage particles. The host cell is light and the phage particles are released. So now the phage which contains the host DNA, that is called as transducing phage. Phage which contains the host DNA is called as a transducing phage. So this transducing phage, when it infects another bacterial cell, when it is infecting another bacterial cell, it will insert its donor DNA. So the transducing phage andre the phage which contains bacterial DNA, some of bacterial DNA. So when this phage infects another host cell, it will insert the donor DNA also. So the DNA from the bacteria, the donor DNA gets included in the recipient chromosome. The donor DNA gets included into the recipient chromosome, and then due to recombination, because through recombination the donor DNA gets included into the recipient cell. So then the bacterium, we say it is transduced because it is now having a part of donor DNA. So this is how the sexual reproduction takes place. Sexual reproduction is not an obligatory step. It is not a very important step in the life of bacteria. But there is exchange of genetic material which can take place through conjugation, transformation, and transduction. See, once I'll repeat conjugation, what is happening? In conjugation, there is direct physical contact between two bacterial cells. The genetic material is exchanged between two bacterial cells through physical contact. So two cells are there. One cell is a donor cell. The second cell is a recipient cell. So the donor cell attaches the recipient cell through the pillars. So the pillars draws the cell together. A tube is formed, which is nothing but a conjugation tube is formed. So once a conjugation tube is formed, from the donor, one strand of the donor strand, the plasmid, it enters into the recipient cell. It is entering into the recipient cell. So in the recipient cell will synthesize a complementary strand to the strand which is passed from the donor. So it contains a plasmid now. The recipient cell, earlier it was F minus, so now it becomes F plus. Now the donor DNA, even that has single strand, other one single strand, so that will also synthesize a complementary strand so that it completes its plasmid. So now both the cells are F plus. So that is conjugation. Conjugation means bacterial cells, the genetic material is exchanged between the bacterial cells through direct physical contact. So there is direct physical contact between the two bacterial cells. The second type of sexual reproduction, second type of genetic exchange, ಅದು ಬಂದ್ಬಿಟ್ಟು transformation. So in transformation ಏನಾಗುತ್ತೆ, donor DNA is transferred to the surrounding medium. Surrounding medium transfer ಆಗಿದೆ. So from the surrounding medium, the DNA is incorporated into the recipient cell. Donor ಮತ್ತೆ recipient cell ಇದೆ. Donor DNA direct ಆಗಿ recipient cell ಟ್ರಾನ್ಸ್ಫರ್ ಆಗಲ್ಲ. That is sending into the surrounding medium. surrounding medium in the that will go into the recipient cell. So the recipient cell will contain the donor DNA. So it becomes a recombination. Recombination takes place. The donor DNA gets incorporated into the recipient DNA. So that recombination occurs. So to explain this, there is a very good example for transformation. I told you the Griffith's experiment. So in Griffith experiment, he studied about Streptococcus pneumoniae. He took two strains of this bacteria. One is called as the R strain. R strain andre cellally capsulilla. The cell is not surrounded by a capsule. The other is a S strain. S strain means the cell is surrounded by a capsule. So R strain is non-virulent. R strain na ನೀವು mouse inject ಮಾಡಿದ್ರೆ mouse will not develop pneumonia. It will be alive. It will not die. S strain na ನೀವು inject ಮಾಡಿದ್ರೆ ಏನಾಗುತ್ತೆ? The mouse will develop pneumonia and it will die. So he studied. He studied, the S strain bacteria. He heat killed the S strain bacteria. Heat kill ಮಾಡಿಬಿಟ್ಟು ಅದನ್ನ ಮತ್ತೆ ಮೌಸಿಗೆ ಇಂಜೆಕ್ಟ್ ಮಾಡಿದ್ರು. So when that is injected, that is the heat killed S strain was injected into the mouse did not develop pneumonia and it was alive. It did not die. So then again he took a combination. He took a combination of both the strains. That is R strain. R strain is nothing but non-virulent. It will not kill the mice. Then is S strain. Heat killed S strain. Even that is also non-virulent. It will not kill the mice. So when these two strains were injected, each two strains, that is R strain ಮತ್ತು heat killed S strain, when he injected into a mouse, in mouse inject ಮಾಡಿದ್ರೆ ಏನಾಯ್ತು ಅದು mouse developed pneumonia and it died. So from the dead mice he observed S cell bacteria, S shaped, S forms he observed. So he concluded that there is certain principle which is transforming the R strain to become S strain. The R form, that is a non-virulent form, is transformed to a S strain, a virulent form, which can kill the mice. So from the dead mouse, he obtained live S strains. So he concluded saying that concluded that there is certain transforming principle is taking place, which is transforming our R strain to a S strain. So that is transformation. The third type here is transduction. In transduction, there is again no physical contact. Transformation also no physical contact. In transduction also no physical contact here, but the DNA is transferred from one bacterial cell to another through a phage virus. Through a phage virus, it is transferred. That is a bacteriophage. It comes and infects the cell. So when it is infecting, it is injecting its DNA into the cell. So when it is injecting, what happens? It will take up the host cell machinery. So it breaks the host DNA, and while assembly of the virus particles, some of the host bacterial DNA can be incorporated into the phage DNA. So when this phage, which is containing the bacterial DNA, it is called as a transducing phage. Phage which contains a donor bacterial DNA, it is called transducing phage. Now when this transducing phage infects another bacteria, then again the same process. It will infect the cell. It will inject its DNA into the recipient cell now. So when it is injecting its DNA, what happens? The phage DNA contains now donor DNA. That donor DNA gets incorporated into the bacterial DNA. So now this bacterium is called as transduced bacterium. Why? Because recombination has taken place. The donor DNA, the DNA from the donor bacteria is incorporated into the DNA of the recipient bacteria. So it is transduced now. So recombination is taken place. So this is the three ways by which the sexual reproduction in bacteria can take place: conjugation, transformation, transduction. Thank you.